2.1 Digestive system and its role in breaking down nutrients. The digestive system breaks down food into absorbable components for energy, growth, and repair. It involves organs and processes working together. Let's explore the key components and their roles. Mouth. Mechanical digestion begins in the mouth through chewing and saliva, containing enzymes like amylase for carbohydrate breakdown. Esophagus. The bolus, a soft mass formed after chewing, is transported from the mouth to the stomach through the esophagus via peristalsis. Stomach. The stomach, a muscular organ, stores and churns food. Gastric juices, including hydrochloric acid and pepsin, break down proteins. Small intestine. The small intestine, a coiled tube, is where the majority of nutrient absorption occurs. The liver and pancreas play crucial roles. Liver. The liver produces bile, stored in the gallbladder, to emulsify fats, increasing their surface area for digestion by enzymes. Pancreas. The pancreas releases digestive enzymes, lipase, protease, amylase, into the small intestine to break down fats, proteins, and carbohydrates. Absorption. Tiny projections called villi and microvilli in the small intestine increase surface area for nutrient absorption. The broken down nutrients, such as amino acids, from proteins, monosaccharides, from carbohydrates, and fatty acids, from fats, pass through the walls of the small intestine and enter the bloodstream. Large intestine, colon. The large intestine absorbs water and electrolytes from indigestible material. Gut microbiota aid in breaking down substances and produce vitamins B and K. Rectum and anus. Indigestible remnants form feces, stored in the rectum until expelled through the anus during defecation. Overall, the digestive system ensures the body receives necessary energy and nutrients for proper functioning and sustenance. 2.2 Mechanical digestion of nutrients. Mechanical digestion involves physically breaking down food through processes like chewing, grinding, and churning. This initial step readies food for subsequent chemical digestion and nutrient absorption. Chewing, mastication. Chewing breaks down food into smaller pieces using teeth and jaws, increasing surface area for enzymatic action during chemical digestion. Saliva, containing enzymes like amylase, aids in carbohydrate digestion. Grinding and mixing. In the stomach, food undergoes further mechanical digestion through muscle contractions, creating churning motions. This breaks down food into smaller particles and mixes it with gastric juices, including hydrochloric acid and enzymes for chemical digestion. Peristalsis. Coordinated muscular contractions propel food through the digestive tract, involving rhythmic waves in the walls of the esophagus, stomach, small intestine, and large intestine. Peristalsis ensures thorough mixing and exposure to digestive enzymes. Segmentation. In the small intestine, segmentation involves localized contractions and relaxations, promoting mixing and churning of food with digestive juices. This aids in contact between food particles and the intestinal lining for nutrient absorption. Emulsification. In the small intestine, emulsification breaks down fat globules into smaller droplets with the help of bile salts from the liver. Bile salts act as emulsifiers, increasing the surface area of fat droplets and aiding fat-digesting enzymes, lipases. Action of the teeth and tongue. Teeth, including incisors, canines, premolars, and molars, play a crucial role in cutting, grinding, and crushing food. The tongue aids in manipulating food during chewing and forming a food bolus for swallowing. Mechanical digestion breaks down food into smaller particles, enhancing surface area for enzymatic action during chemical digestion. 2.3 Chemical digestion of nutrients. Chemical digestion involves breaking down complex food molecules into simpler ones through enzymes and digestive substances. Key aspects include stomach. Gastric juices in the stomach, containing hydrochloric acid and pepsin, create an acidic environment. Hydrochloric acid denatures proteins, and pepsin breaks them down into smaller peptids. Pancreas. The pancreas secretes enzymes into the small intestine. Pancreatic amylase digests carbohydrates, pancreatic lipase breaks down fats, and pancreatic proteases further break down proteins. Liver and gallbladder. The liver produces bile, stored in the gallbladder, aiding in fat digestion. Bile emulsifies fats, increasing the surface area for enzymatic action. Small intestine. The primary site for chemical digestion and nutrient absorption. It receives pancreatic enzymes, bile, and produces its own digestive enzymes. Maltase, sucrase, and lactase break down disaccharides, while peptidases break down peptids. Lipases act on emulsified fats. Absorption. After chemical digestion, smaller molecules like glucose, amino acids, fatty acids, and glycerol are absorbed into the bloodstream or lymphatic system. These nutrients are transported to cells for energy production, growth, and metabolic processes. Chemical digestion is crucial for breaking down complex food into basic building blocks, facilitating absorption and utilization by the body. Enzymes and digestive substances play a key role in ensuring efficient nutrient extraction from food. 2.4 Carbohydrates absorption of nutrients. Carbohydrate absorption involves the uptake of simple sugars from the digestive system into the bloodstream for body utilization, mainly occurring in the small intestine. Key steps include digestion of carbohydrates. Complex carbohydrates like starches and disaccharides are broken down into simple sugars. Salivary amylase in the mouth initiates starch breakdown, and pancreatic amylase in the small intestine continues the digestion of polysaccharides and disaccharides. Pancreatic amylase. Once food enters the small intestine, pancreatic amylase, secreted by the pancreas, is released into the duodenum. Pancreatic amylase continues the digestion of polysaccharides and disaccharides into smaller sugars, such as maltose, sucrose, and lactose. Brush border enzymes. Brush border enzymes, maltase, sucrase, lactase, on intestinal cells further break down disaccharides into monosaccharides. Maltase breaks down maltose, sucrase breaks down sucrose, and lactase breaks down lactose. Absorption into intestinal cells. Monosaccharides are transported across intestinal epithelial cells by specific transporter proteins on the apical membrane. Glucose and galactose use sodium-dependent glucose transporter 1, SGLT1, while fructose uses facilitated diffusion through glucose transporter 5, GLUT5. Transport to the bloodstream. Inside enterocytes, glucose, galactose, and fructose are transported into the bloodstream via glucose transporter 2, GLUT2. This allows their exit from enterocytes into capillaries of intestinal villi. Monosaccharides are then transported through the bloodstream to tissues and organs for utilization. Carbohydrate absorption is vital for delivering glucose, the primary energy source, throughout the body. Proper absorption is crucial for maintaining blood glucose levels and providing energy for cellular functions. 2.5 Protein absorption of nutrients. Protein absorption involves breaking down dietary proteins into amino acids for utilization by the body, primarily occurring in the small intestine. Key steps include digestion of proteins. Protein digestion starts in the stomach, where pepsin breaks down proteins into smaller polypeptids. Further digestion occurs in the small intestine. Pancreatic enzymes. Pancreatic enzymes, trypsin, chymotrypsin, carboxypeptidases, in the small intestine further break down proteins into smaller peptides and individual amino acids. Brush border enzymes. Brush border enzymes on intestinal cells complete the digestion of peptids into individual amino acids. Aminopeptidases and dipeptidases further break down peptids. Absorption into intestinal cells. Amino acids and small peptids are transported across intestinal epithelial cells by specific transporter proteins on the apical membrane, including sodium-dependent amino acid transporters and hydrogen-coupled peptide transporters. Transport to the bloodstream. Inside enterocytes, amino acids are transported into the bloodstream through specific transporter proteins on the basolateral membrane, like sodium-independent neutral amino acid transporters, e.g., LAT1 and LAT2, and sodium-dependent neutral amino acid transporters, e.g., ASCT2. Amino acids enter the capillaries of intestinal villi, transported via the bloodstream to tissues and organs for utilization. Protein absorption is essential for providing amino acids necessary for synthesizing new proteins and supporting various biological processes. Proper absorption ensures the availability of essential amino acids, supporting tissue growth, repair, and maintenance throughout the body. 2.6 Fats absorption of nutrients. Fat absorption involves breaking down, emulsifying, and absorbing dietary fats into the bloodstream for body utilization, primarily occurring in the small intestine. Key steps include emulsification. Bile salts from the liver, stored in the gallbladder, emulsify fats in the small intestine, breaking larger droplets into micelles. Lipase action. Pancreatic lipase, released into the small intestine, breaks down triglycerides into fatty acids and monoglycerides, allowing penetration of micelles for enzymatic action. Micelle formation. Products of fat digestion combine with bile salts to form micelles, transporting fat digestion products to the surface of intestinal epithelial cells for absorption. Absorption into intestinal cells. Micelles interact with the brush border of intestinal cells, and fatty acids, monoglycerides, and other fat-soluble compounds diffuse into enterocytes, where they are reassembled into triglycerides. Formation of chylomicrons. Reassembled triglycerides combine with cholesterol, fat-soluble vitamins, and proteins in enterocytes to form chylomicrons, transporting dietary fats through the lymphatic system and into the bloodstream. Transport through lymphatic system. Chylomicrons enter lacteals, specialized lymphatic vessels, and reach the bloodstream via the thoracic duct, connecting to large veins near the heart. Utilization and storage. In the bloodstream, chylomicrons deliver dietary fats to tissues for energy or storage in adipose tissue for future energy needs. Fat absorption is vital for providing energy, insulation, and essential fatty acids, ensuring dietary fats are broken down and transported for utilization or storage, supporting various physiological functions. 2.7 Vitamins and minerals absorption. Vitamins and minerals, essential micronutrients, are absorbed through diverse mechanisms based on their properties. An overview of absorption. Water-soluble vitamins include vitamin C and B-complex vitamins. Absorbed in the small intestine through passive diffusion or facilitated transport. Absorbed directly into the bloodstream, excess amounts are typically excreted in urine. Fat-soluble vitamins include vitamins A, D, E, and K. Require dietary fat for absorption, transported through the lymphatic system. Incorporated into micelles during digestion, absorbed by passive diffusion into intestinal cells, and then packaged into chylomicrons for transport into the bloodstream. Minerals. Calcium and magnesium. Absorbed in the small intestine through active transport mechanisms. Vitamin D enhances calcium absorption by increasing the expression of calcium transporters. Iron. Absorbed in the duodenum and upper small intestine. Absorption involves duodenal cytochrome B, DCYTB, dependent reduction. Inside intestinal cells, iron is stored as ferritin or transported into the bloodstream via ferroportin. Zinc, copper, and other minerals. Primarily absorbed in the small intestine through active transport mechanisms. Specific transporters facilitate absorption into intestinal cells. Once inside cells, minerals can be stored or transported into the bloodstream. Vitamin and mineral absorption processes are diverse, reflecting their distinct chemical properties and necessities for various physiological functions in the body. 2.8 Water and electrolytes absorption. Water and electrolyte absorption, crucial for fluid balance and electrolyte homeostasis, primarily occur in the small intestine. An overview of these absorption processes. Water absorption. Mainly through osmosis, driven by an osmotic gradient established by solutes. Water moves from the intestinal lumen to the bloodstream through enterocyte cell membranes. Absorbed water is transported through intestinal cells into the bloodstream for distribution. Sodium absorption. Primarily through active transport, particularly via the sodium-glucose cotransporter, SGLT1. Glucose and amino acids co-transport with sodium into intestinal cells. Sodium is transported out of cells into the bloodstream by sodium-potassium ATPase pumps, maintaining the concentration gradient. Chloride absorption. Passively absorbed alongside sodium, following the electrical potential established by sodium transport. Chloride ions move paracellularly or transcellularly depending on the specific small intestine segment. Other electrolytes, potassium, calcium, etc. Absorption occurs via specific mechanisms involving active transport, passive diffusion, or facilitated transport in the small intestine. Regulation involves factors like hormones, transport proteins, and dietary intake. Colonic water and electrolyte absorption. In the colon, absorption occurs through passive diffusion, osmosis, and active transport mechanisms. Supports the final concentration of stool and helps maintain fluid balance. Overall, the small intestine is the primary site for regulated water and electrolyte absorption, ensuring proper hydration and electrolyte levels for maintaining overall fluid balance and homeostasis in the body.